Welcome all of yours. So today I will present some data and results about sandfly-borne viruses of medical importance, only the one that are human pathogens. So first, just let's talk a little bit about the arthropod-borne viruses that are transmitted by sandflies. As you can see, they belong mostly to three different families of viruses. One is Shanginola. Some viruses are in the Rhabdoviridae family, and as you can see from the map, they are mostly in the New World. And the last one is the Phenuiviridae family, in which we have the genus Flebovirus. And the virus we are going to talk today are mostly around the Mediterranean basin in the New World. So, as you can see in this map, we are going to talk about three different viruses, and this is the distribution of these viruses based either on serological data or molecular PCR data or isolation of viruses. So this data has been gathered for 70 years now. So it's a cumulative effect. But what is important to see is that basically all the countries bordering the Mediterranean Sea are exposed to the circulation of at least one of these viruses. The important point that is quite new recently is that the climatic change is going to have a direct and massive impact on the distribution of the vectors, and hence the distribution of the viruses transmitted by this vector will be impacted. So we don't know yet, but what we have seen so far is a migration of the sandflies and the viruses associated with these sandflies with a south to north gradient. So basically, the north part of Europe will be and is already more affected than it was several decades ago. The second point that is important is that some of these viruses can be transmitted by different species of sandfly. So basically, the specificity between the virus and the sandfly species is not so strict and stringent, which is also important to be taken in consideration. So flamoviruses, very briefly, they can be transmitted by mosquitoes, they can be transmitted by ticks, and the one that we are dealing with today are the ones that are transmitted by sandflies. And they are the majority, we can say. So we'll only talk about sandflies. So short introduction, also you probably know that better than me, is that sandfly first, they are very small. 1/3 of a normal mosquito. By normal, I would say Aedes or Culex mosquito. They are hairy. They are very mobile. So basically, they take their time to settle and to bite. They are totally silent. You cannot hear them. So you're not disturbed by sandflies during the night. And they are mostly nocturnal, but some can also bite during daylight. What is important is that their density is varying according to the climate in which they are circulating. And as you can see, the period during which you can witness sandfly circulation is very large, is almost 8 months per year, and probably it will increase with the climate warming or climate change. So we can expect that this time span will expand a little bit. So far, what we can see is that during the last 20 years, we had cases of Toscana virus, and we will, I'm not revealing this virus, but we had human cases in the second part of March, and the latest case we witnessed were in November, and I'm talking about southern France. So as you can see, even in southern France, the time span is very large. Oops. Okay, so flaviviruses, they belong to the order Buniaphilorales in the family Pheniviridae in the genus Flavivirus. They are RNA viruses. They have 3 segments. And even so, the taxonomy has drastically changed over the time. Basically, the viruses have not changed. They are still the same. It's just the way we classify them that has changed. And as you can see now, we have 63 species and in the genus Flavivirus, and the family Pheniviridae, it's more than 350 species. So they all are possessing 3 segments, so they are prone to reassortment and also to recombination. So these viruses can infect a large variety of vertebrates, and their cycle involve arthropods, in this case, sandflies, and various vertebrates. Also, in most cases, the animal reservoir is not known if it does exist. So there is not a clear evidence that for these viruses there is an obvious and a demonstrated vertebrate reservoir. But what is important from this schematic is that the transmission of the virus can occur within the sandfly population through venereal transmission between male and female, transversal transmission, the female to the eggs, and also, most likely, but because the vertebrate host have been identified, but we don't know now. I mean, we know that they can be infected, but we don't know if they can reinfect sandfly after being bitten. So does this human, dogs, bovine, cattle, equine, and so on, do they play a role, or are they only dead-end hosts? We know that for most of them, they are dead-end hosts. They do not play a role in the cycle. So maybe small vertebrates are playing a role, or birds can be playing a role, but there is no firm demonstration for this now. So this is a very basic phylogenetic tree. And as you can see, in the viruses that are transmitted by sandflies, we can distinguish 3 different complexes. The Naple complex, the Sicilian complex, and these 2 complexes include all the human pathogen, and the Saliabat complex, in which there is a growing number of viruses. I think now there are more than 10 different species of viruses, but none of them have been demonstrated as human pathogens. So what is interesting with sandfly is that they can cause these viruses. They can cause sandfly fever, and sandfly fever has been one of the first arboviral diseases to be recognized. Even so, at this time, there were no firm knowledge about the nature of the virus and the causing agent was the virus. But basically, there was a very old link between sandfly fever and the presence of sandflies. So basically that when you are bitten by sandfly, you can show an infection which is mainly characterized by fever. So this fever has been named like phlebotomys fever, papatachi fever, because of phlebotomys papatazi that was highly dominant at the mid 20th century. 3 days fever, because usually it lasts only 3 days. So I have. Okay. Or sandfly fever. As you can see, the first demonstration that sandfly fever was caused by a filterable agent that was later called the virus was at the beginning of the 20th century. So it's one of the first viral diseases to be documented. Afterward, what is interesting is that sandfly fever has been mainly a military problem. There are many papers and reports of outbreaks, epidemics in different parts of Europe and Asia during World War I and also during World War II. So what was worrying the physician and the military and the people is that the troops were healed for 3 days and were totally incapacitated. So basically there was a big military problem. It's why most of literature is military literature early. So what was reported in the literature at this time was the symptoms. The symptoms were people having fever, headache, heavy headache, muscle pain, photophobia, retroorbital pain, and incapacitation. You have to recall that at this time there were no aspirin, there were no ibuprofen. So basically, when you were sick with a viral disease, the only thing you could do is to wait until it passes or until you die. So there is no symptomatic treatment in this time. The important point is that it was reported large epidemics. When I say large epidemics, it's large at the scale of the country. So the Greek epidemic is probably almost 100,000 cases. As the one in Ethiopia, it's 10,000 of cases. Cyprus, which is a small island, it's hundreds of cases. Serbia, it's also 10,000 of cases. So basically, it's really massive attack and very large outbreak reported at this time. So the interesting point is that we have two different viruses that can cause this disease. And as you can see, you have two different distribution of these viruses, the sandfly fever Naples virus and the sandfly fever Sicilian virus. Basically, they both occupy same country in the Mediterranean basin. Also, the evidence for the two viruses may be a little bit divergent in terms of geographic distribution. So what is interesting is that in 1971, a new flavivirus was discovered in central Italy. It was called Toscana because it was discovered in Tuscany. And it was discovered by the group of Paola Verani from Phlebotomys perniciosus and Phlebotomys perfilievi. If you recall what I said before, Naples and Sicilian had been isolated
mainly from Phlebotomus papatazi, was highly dominant around World War II, World War I, and World War II. What's interesting is that Toscana is antigenically related, almost distinct, to Naples virus. The second important point is that the link between Toscana virus and a human disease has been done only 16 years after the virus was discovered. So basically, this virus was considered as a flavivirus without any human harming or human disease, until it was reported first, I think, maybe a little bit earlier in Germany, but after in the US about Charlie Kalischer. So the first case was a case of disease occurring in the US, and the patient was returning from a stay in Italy. Afterwards, there were some studies initiated by a Paola Verani group in Italy. And as you can see, in 1991, there were some large number of cases documented, and all of this for almost 15 years, all the cases were reported in Italy, where in Tuscany, Toscana virus can cause 80% of summer meningitis in this time. So it's a major cause of human infection. So as you can see here on the table, we still have the Naples complex, and basically in the sandfly fever Naples complex, you also have Toscana virus that is distantly related, but related to Naples virus. Still the Saleh-Abad complex with no virus pathogenic for humans, and the Sicilian complex. So if we summarize the symptoms associated with these two viruses, Sandfly Sicilian and Sandfly Naples, first point, both viruses can cause strictly the same disease. The disease is sandfly fever, papatachi fever, 3-day fever, and it's characterized by a febrile illness, more or less incapacitating. Toscana virus, the newly discovered virus, is exactly causing the same disease, but the difference is that in some cases, it can cause meningitis and central nervous system infection with encephalitis and also peripheral nervous system infection. So it seems that Toscana specificity is that it has a attraction for the nervous system of humans and causing something that is a little bit more severe than these cases. So after 1971, what we have is 3 different viruses. The 2 first, Sicilian and Naples, are mostly, or are believed to be mostly, transmitted by Phlebotomus papatazi. They cause the same disease, sandfly fever. Toscana, transmitted not by papatazi, but perniciosus and perfilievi, and it causing also sandfly fever, but it also can cause more severe disease like meningitis, encephalitis, and peripheral neurological infections. So if we look back in the literature and we focus on Toscana virus, what we can see is, and this is PubMed screening, is that there is only 330 research and review articles from 1982, the first report, to now. So it's very limited, and it has to be clear that this virus has a lot of things to reveal because there is limited knowledge because of the number of cases that were reported are not so high. So the important point is that, also, there is clear evidence that Toscana is causing human disease, and it's causing human disease largely
it remains poorly studied. And what we can see after 2000, as I said, before 2000, there was only cases reported in Italy, and in fact, one case in Portugal. So the end of the 20th century, the beginning of the 21st century, there is evidence, accumulating evidence that Toscana is not restricted to Italy. It's also in Spain. And then it was looked for in France a little bit later after Spain. And most of this work in France was done in my group and also with the National French Center for Arboviruses. And the conclusion of this is that Toscana virus is in the top 3 causes of meningitis in Italy, France, and Spain. Of course, these meningitis are restricted during the activity of the sandfly that it transmitted the virus, and it's basically from late March to November with a peak during the warm period, which is June, July, August, September. So basically the 4 months with maybe an even bigger peak in July and August. And more investigations have been conducted by different groups. And as you can see, outside of Italy, after France, Portugal, and Spain, there were reports of cases in Turkey, in Greece, in Elba Island, in Morocco, in Tunisia, Algeria. So the southern part of the Mediterranean basin is also touched. Croatia, Corsica, which is the French island, Romania, Germany recently, Portugal, Libya. So as you can see, the picture is enlarging very rapidly. This is basically a map that was drawn, I think, something like 10 years ago with the most recent data. Another thing is that there are 3 different genotypes of Toscana virus. Most of the first cases that were reported in Italy are caused by lineage A because it was the first strain to be isolated, but lineage A is not restricted to Italy. You can see also in Turkey, in Tunisia, in France, in Algeria. After you have lineage B, which is more in Spain, in France, which looks like lineage A is lineage B is Western Europe. Lineage A is a little bit more Eastern within Western Europe. France has 2 lineages. Turkey has 2 lineages. Croatia has 2 lineages, including lineage C, that has never been isolated. And the Balkans have also lineage C. So importantly, there is no link so far between the lineage and the severity of the disease or the length of the disease or specific clinical manifestation. Another point that is important is that progressively what we have seen is that Toscana, even though it is mainly transmitted by Phlebotomus perniciosus and Perfilievi, might be, and there are accumulating evidence that it could be transmitted also alternatively by other sandflies like Longicuspis, Sargenti, Toby, Neglectus, and maybe Sargentomia. There is one report, but there is no clear evidence for Sargentomia. But the other Phlebotomus, it seems that they can transmit the disease. This is where we have conducted, and it has been conducted, a seroprevalence study. So briefly, what is interesting, is that if we compare Western Europe, the seroprevalence are much lower in the general population. As you can see, Spain, Portugal, France, Italy, it's less than 20% in all the studies. the prevalence is much more higher in the southern part of the Mediterranean basins, like Morocco, Algeria, Tunisia, with at least 30% of the population exposed having antibodies against Toscana, and also the Balkans. So basically, the expansion of Toscana infection might be more rapid in the Balkans and Central Europe than in Western Europe. Even so, the climatic change might modify a little bit this tendency. This is just one map. I will go fast because we were addressing the question of the incubation period for the infection by Toscana virus. As you can see on the left, you have the estimation from the reference in the literature. It's that basically what was reported in papers. So from 1996 to 2020, by scrolling 22 different papers with mention to incubation period, as you can see, the incubation period is usually short, 2 days, 3 days, 2 days, 7 days, and none of these was really demonstrated. So what we wanted to do at this time is trying to identify and to better understand this incubation period. And what we did is to perform studies by scrolling the literature and selecting the papers where we could have information about this period. So basically, what we had to do is to take cases of TOS-infected travelers that were returning in non-endemic homeland to be sure that they had acquired the infection abroad and not in their country. So we did the analysis of this. We selected 24 different cases that were clearly reported. And as you can see from the map, most of the cases, I mean, a large majority of cases were returning in northern country where Toscana is not present. So in Germany, in UK, in Sweden, in France, northern France, not southern France. And they were coming from southern part where the Toscana virus is endemic and where it causes summer disease. So what is interesting is by scrolling, analyzing these 24 cases, we could determine that the mean incubation period was much longer than believed and reported in the literature based on approximation. So we define that for neuroinvasive cases only, and I have to be clear that we are only talking about neuroinvasive cases. If these neuroinvasive cases, meningitis, are the incubation is the same for the normal fever is unknown at this time. So for neuroinvasive cases, which were the case for these 24 cases, the median length is 12 days, which is much higher than for other arboviruses, because the length of incubation period for other arboviruses, as you can see, goes to 2.6 to 5.6 days. And this is demonstrated data. So it is important, and it also raised the question of how long you should suspect Toscana infection when somebody is coming back from an endemic area. And as you can say, from the 24 cases, you can at least expect incubation after 2 weeks, maybe a little bit longer if you increase the number of cases. But so far, this is what we have in being able to demonstrate. So this is very rapidly a report on the comprehensive review that we did with Nasle and Carol by reviewing almost 1,400 cases of infection. As you can see, most of the cases are reported in Italy. And after you have a decreasing number of cases, What is interesting is that at this time, we don't have cases reported from human cases from Morocco, from Libya, from Egypt, from all these countries in the Central Europe and the Balkan, and all these countries, there are not cases, but basically you can expect that the virus is circulating there, and there is no reason why it should not be present. So basically, we expect that Toscana virus is widely distributed in the all around the Mediterranean basin. So you will have access to this slide. So I'm not going very. This is basically what we reported and analyzed. Is that most of the case present with sandfly fever, and you have a significant number of cases with neuro-invasive manifestation, which is normal because most of these cases have been reported after hospitalization, and so only the most severe cases are hospitalized. So we don't know the full picture of the infection in the mild cases. Okay, so you will have this data. So at this time, so far there is no prevention or therapeutic antivirals for Toscana virus. What is important to remember is that Toscana virus is one of the most frequent arbovirus in Europe and in France, and that more than 500 million people are at risk of contracting Toscana virus infection because they are exposed, they are living in exposed country. And I just would like to finish with this, is that we are talking about true human pathogen, and they still deserve serious study to better understand the disease they are associated with. So, until recently, the reason for they remain neglected, is that there was a clear lack of diagnostic arsenal, and with very few serological kits and no commercially available molecular data. So the situation has a little bit changed now because at least for Toscana, you have an ELISA test that has been commercialized by the Italian company DS for almost 20 years now. There are also Euroimmune that is commercializing a serological kit for Toscana and for Sandfly fever Sicilian, which is based on immunofluorescence, and it's a very nice kit. And for the molecular testing, you now can have access to reagent that can detect at this moment Toscana virus through the EVA catalog. These tests have been validated fully. Oops. They are produced in the platform that is ISO 13485, so they have high quality grading. They are commonly used and routinely used in the National Reference Laboratory for Arboviruses in France, and you can get them from this link. And of course, they are commercially available, but there is also possible to develop collaboration. And in this case, we can provide the test for free. So now, what is important is because we have the tests that are available, it's to try to copy what Italy has done, because in Italy, Toscana virus has been included in the first line screening in the surveillance of arboviruses. It might be the reason why Italy is the country where the largest number of cases are reported. And basically the same should apply in countries with significant seroprevalence rates that demonstrate that the virus is present and circulating. There is a clear, very simple option is to use the West Nile surveillance for Toscana study. It's cost-effective study, and it can provide rapid results within 2 or 3 years. The second option is to conduct cohort studies on fever with unknown origin during summertime. These type of cohorts can be screened retrospectively with the diagnostic assay that are now available, but also can be conducted prospectively. And we engage that this fever of unknown origin would target Toscana virus and Sicilian virus, for which the molecular tests are going to be available on the EVA platform during this year, 2025. So this could be implemented in all the clinical virology, microbiology labs that have serological capacity and RT-qPCR capacity. And if you're interested in creating a consortium to try to raise fund for external funding, just drop me a mail and I will be happy to answer you and to try to create a community around this. So I've been a little bit longer than expected. So this is my last slide. And now, if you have questions, I will be happy to take them.
Thank you so much.